Today is a busy day, looks like, because of Science Day celebrations as well as the related other events.
Right.
Yeah, so something came up all of a sudden for him. And
So now it is live
yeah, okay.
on YouTube.
Yeah, oh, okay.
We'll just start in 1 minute. Good morning. A very warm welcome to all of you to the National Science Day celebrations of ICO Tirupati. Today we'll have two sessions, and in the forenoon session we'll have talks from two invited dignitaries, Dr. Shahid Jameel and Shri Ramji Raghavan. In the afternoon, we'll have student presentations as part of the national virtual science fair. Before we can actually start the event, I would like to inform you that Professor Ganesh is unable to join us today due to a meeting that came up with a very short notice today. So he conveyed his apologies and also extended his greetings to all of us. And we have Professor B.J. Rao, Dean Faculty, who will give his opening remarks for the Science Day events and also introduce the speakers. Sir, over to you.
Friends, welcome you all to ISER Tirupati on National Science Day celebrations. 28th February 1928 was a very auspicious day in Indian history, which is when Sir C.V. Raman and Krishnan actually observed Raman lines for the first time in the world in Indian Association of Cultivation of Science. Very important milestone that the country celebrates every year as Science Day. And today at ISER Tirupati, we are celebrating Science Day event by hearing two stalwarts in two different domains of knowledge. Shahid Jameel in the domain of virology and Ramji Raghavan in the domain of how to do creative education in the country and in the world at large. These are two different colossus personalities that we have today. And therefore, I would feel it's my special privilege to invite them to our amidst in the virtual world and hear them. I would first introduce my very good friend Shahid Jameel, a very long associate in Indian biology. Shahid did his early education in Aligarh Muslim University, followed by IIT Kanpur. And then he went into this domain of regulation in virology, in viral systems. And he got his early upbringing in the virology domain in the University of Colorado Health Science Center, Denver. He finished his postdoctoral research there and returned to India to set up a very renowned virology lab, first time in the country, at ICGEB, International Center for Genetic Engineering and Biotechnology, that he did in 1988. He was the virology group head. He did pioneering work in virology and put India on the map of virology research in the world. He was recognized by many laurels in the country, foremost being Shanti Sarup Bhatnagar Award. He was elected fellow of all national academies. He mentored several young virologists in the country. He published very high-profile papers from India, and therefore, he was one of our very cherished virologists from ICGEB. After finishing the stint from ICGEB, he took up a very interesting responsibility as Chief Executive Officer, a science administration position in Wellcome Trust DBT India Alliance program, a very interesting pan-India program whereby Indian biology talent is recognized, nourished, handheld through very open, competitive funding process. He was the CEO for Wellcome Trust DBT India Alliance for about 7 years. And he now made a transition into real hardcore undergraduate education at Ashoka. He is the director for Trivedi School of Biosciences. And I'm sure he's enjoying teaching large number of students at Ashoka. And I know he's a very interesting teacher. He is also an avid photographer for details. You can talk to him about the intricacies of photography. You will learn a lot about him. So Shahid, welcome to ISER Tirupati. But before I give the podium to you, I also want to introduce Ramji Raghavan sir, a very interesting person that I have met in the recent past. Ramji Raghavan sir is actually a business entrepreneur, an MBA from London Business School, spent time at the International Institute of Social Sciences, Netherlands, and then Delhi University. So his early upbringing, I suppose, was in the intricacies of how to set up business models. And then maybe using this particular backdrop, he thought maybe one could do innovative things in India, in educational India. So he founded this organization called Agastya International Foundation in 1999. It's a non-profit organization. In this organization, the main agenda is to impart creative, hands-on experience to young students, especially from underprivileged category. Very large number of underprivileged category students who are extremely talented, creative, do not have direct exposure to the methods of creativity. And this is what Agastya Foundation is trying to create, is trying to provide. In fact, by now, they have given hands-on experience to about 12 million underprivileged students in the country, a large number. He is an expert in scaling up of models. It's very clear. And 250,000 teachers have been handheld in terms of new teaching methodologies. Agastya Educational Foundation has innovated interesting methodologies such as mobile science labs, not science labs on mobiles. These are mobile science labs which actually move in the country, in the hinterlands of the country, and reach out to the underprivileged, school children with the labs on bikes, that is labs on bicycles, and interesting methodologies where peer-to-peer learning can happen. A very interesting area where we have not really tapped too much into the potential of peer-to-peer learning, which is what Ramji Raghavan sir is trying to do. So there is a lot that I can tell about Ramji Raghavan sir, but I do not want to intrude into the time of this Science Foundation Day, Science Day. But I certainly want to tell that he is intimately connected with many governmental committees. He is a member of Central Advisory Board of Education, Government of India. He is also a governing council member of an interesting foundation called Marico Innovation Foundation, who actually do a lot of nurturing, hand holding, and sustaining of startup companies by giving them hands-on ideas of how to unclog the bottlenecks during startups. So you can see that Ramji Raghavan sir is a very interesting person trying to help education through real innovative methods. So we have two interesting personalities today, Shahid Jameel ji and Raghavan ji, and I think all of us are blessed with their experience, knowledge, and reach out that we want to benefit from. And I want to leave the podium to both of them, first to Shahid Jameel sir, and then afterwards to Ramji Raghavan sir. Thank you very much for giving me this opportunity and welcome you all once again. Thank you. Shahid, you can take over and start.
Yes.
Yeah.
Thank you very much, BJ, for that extremely generous and kind introduction. Uh, it's interesting what you said about mobile labs. Yes, in today's day and age, a mobile lab really means lab on a mobile.
Yes.
And, we are all sort of learning this in our daily lives on how to handle education online and uh, the pluses and the minuses of online education. Nevertheless, that's the best we have at this time. And while I would have much preferred to to be able to do this uh, in person in Tirupati, uh, we really have to work with what we have, and therefore just do this online. Let me just give me half a minute to get going.
Sure.
My system has slowed down a little bit, uh, and you can be in the best of places and still have these problems.
Yeah, that's the nature of the technology.
Yeah. Uh, just a minute. Just give me a minute. I'll possibly have to reopen my slides. So just give me a minute.
Sure, sure, sure.
Okay. Set to go. All right. I hope you can see my screen, and I'll
Correct.
put it
Yeah.
on
Go to full view. Yeah.
on the
You're
view
all set.
mode.
Yeah.
I will be talking about COVID-19 in India. All of you know what COVID-19 is, so there is really no need to prime you to that. But there's a lot that has happened over the past 12 months or so. If you recall, in India, the first case was on 30th of January 2020, and that really seems like a lifetime ago. But let's look at the situation in the country. And so, what I would be doing in this talk is to really outline the pandemic in India, look at the numbers a little bit, and then ask this question: how many infections do we really have? There's also concern that we may be looking at a second wave. Are those concerns true? Are they not true? What might possibly be the reasons or that we are seeing in the spike, especially in Maharashtra, that's happening right now? I'll get into my comfort zone then and talk about the biology of this virus, especially how it relates to genomic diversity, the mutations that are arising, how those mutations impact vaccines that are being developed, and then just give you an overview of these things. All right. So if you look at the global pandemic, there are over 111 million cases, confirmed cases around the world. But confirmed cases simply means people who have been tested and found to have the virus. There is a big chunk of people who don't get tested simply because they remain asymptomatic, and therefore they're not really counted in this. And that happens in every country, not by design, but simply by the nature of the infection and disease. And that has happened in India as well. But India at this point has roughly 10% of the world's confirmed COVID cases, about 11 million cases so far. Uh, as far as the deaths from COVID are concerned, US has already gone over 500,000 deaths, 500,000 deaths. India is number 4 in the world in terms of number of deaths. We are close to 156,000 people who have perished in this outbreak in our country. And as you will see, the spread of the virus is really global. Every country has really been impacted, both by the disease, but also by the socioeconomic aspects of this infection that's going around. Looking at cases in a little more detail, so as I said, India is pulling roughly 11 million cases so far, 10% of the global cases, 156,000 deaths. But if you look at the case fatality ratio in India, that turns out to be roughly 1.4%. And at this time, our recovery rate is a little over 97%. So things went really quite badly in August and September. But then after mid-September, the caseload started going down, and we are actually in a fairly comfortable position right now. But having said that, that brings in a level of complacency, and that may cost us dear in some places, as we are seeing now. Uh, so India's case fatality rate is roughly half of that of the global average. The global average stands about 2.4, 2.5%. So that's something else to be understood. Why, considering the poor healthcare system that we have, why our case fatality ratio is so much better than rest of the world? Well, the easy answer to that you may have heard is that we're not counting deaths properly. That's possible, that even in the best of times, India, the reporting structure is such that we miss 80% of deaths because they don't happen in an institutional setting. Nevertheless, I think the reasons for this are more complex and have to do uh, both with the young population that we have in the country. So, for example, 50% Indians are below 25 years of age, 65% Indians are below 35 years of age. So, age is in our favor. The other biological hypothesis is that since we are exposed to so many other infectious diseases, perhaps our innate immune system is more primed to handle viruses like this, and it controls the virus in our nasal cavity and in our throat before the virus proceeds to our lungs. And as you know, it is the pneumonia, the effect when lungs are affected, that causes severe disease and fatality. So there are multiple possibly reasons for it. Look at in time what has happened over the last 12 months. India started going up around May or June, actually May, and then peaked around mid September. And after this peak arrived in mid September, there's been a downfall in the curve, even survived the Dasara Diwali festive season, survived Bihar elections, and didn't really see a spike. But nationally, there appears to be a little upturn in cases now, and that's really largely because of cases that we are seeing coming from Maharashtra and Kerala. At this time, 80% of cases, daily cases that are being reported in the country are essentially coming from two states, that's Maharashtra and Kerala. While Kerala has remained fairly stable over the past few weeks, Maharashtra is spiking, and certainly it's the Vidarbha region in Maharashtra that is spiking the most at this point. All right. So this is what I was talking about. The caseload in India seems to be rising, and this is really amplified for the month of February. And you will see that after some initial up and down, cases have continued to go up in the country from a low of roughly 9,000 cases per day to now roughly 15,000 cases per day. And this peak is really being contributed. Well, it's not a peak yet. This increase is largely contributed by Maharashtra, which seems to be going up. And even in Maharashtra, the areas that are going up are in the Vidarbha region, Akola, Amravati. You know, this is the area, Nagpur. This is the area that's going up. Small increases are also being seen coming from Madhya Pradesh and Punjab. But if you look at the numbers are really around 350, 300 to 350, whereas Maharashtra is pulling roughly 7,000 cases on a daily basis. Kerala, as I said, has remained high but stable. And the government of Kerala is really focusing more on mortality rather than on increasing caseload. So the mortality in Kerala continues to be very low. Uh, this plots the number of active cases on a daily basis, the blue line. And as you will see, the number of active cases came down, have continued to come down since mid-September, reached a low around mid-February, and then have started going up again. So currently we have roughly 150,000 active cases on a daily basis. Uh, but it is the bottom curve that is causing a bit of worry to people. And there are two lines here. The dark line is the new cases, and the light colored line is the recovery. So if you see somewhere around this point, somewhere around mid February, the two curves have intersected. So now we are actually having more new cases coming on a daily basis than the number of people that are recovering, which means that the active case numbers are going up. So this is something that is worrisome at this point. So let's ask this question: how many infections are there in India in real terms? So on 22nd of September, we had 5.6 million confirmed cases in the country. At that time, ICMR released their second sero survey, which showed that 6.6% people across 70 districts of India, a test population of about 28,000 people, uh, had antibodies. Now, if this is representative for the country, then that amounts to roughly 91 million people exposed to the virus around the third week of September. So if you divide this 91 million by 5.64, which was the actual caseload, you come with a factor of 16. So the idea is that we were really undercounting by a factor of about 15 or 16. The third ICMR Sero survey was released a few weeks back. This showed that 21.4% of adults in the country and 25.3% of 10 to 17-year-olds have antibodies. So roughly, let's say 22% people have antibodies. So if you translate the 11 million cases in India as of now, and this data is a few days old, uh, we have roughly 175 million possible cases in the country. But this is all over India. If you look at some cities, so for example, if you look at Delhi, in August, Delhi have already had about 30% seroprevalence. The new Delhi Sero survey after this has shown 57% people on average in Delhi have antibodies. Mumbai is sort of the same thing. Pune is the same, is pulling the same numbers. So there are locations in India, mainly big cities with dense populations, that are already somewhere between 50 to 60% exposure. But there was another interesting report from this testing company called ThyroCare. In July, ThyroCare released results from 60,000 antibody tests that they did across 600 uh, you know, pin codes around India. And if you look at that figure, they were showing 15% positivity in July. That translates to roughly 200 million people. On 19th August, ThyroCare repeated that, this time with 2 lakh tests, and found 24% positivity. And 24% of India is roughly 330 million people. So the point that I'm trying to get across is that while our confirmed case load is roughly 11 million, we possibly have somewhere in the range of about 3 to 400 million infections in the country. So keep that number in mind, because that also has a bearing on uh, why numbers have gone down consistently since September, and why we are not really seeing the kind of spikes across the country, because people have really stopped wearing masks. People, everything is open, back to normal. People are interacting with each other as if nothing had happened. Now let's go to the biology of the virus and its genomic diversity. Now the SARS coronavirus is a member of the coronavirus family. Uh, these are single-stranded RNA viruses. They have a large RNA, roughly 30 kilobase, and it's the largest RNA virus genome known to us so far. Uh, Normally, RNA viruses are subject to a lot of mutation, and that happens because the enzyme that replicates the RNA genome has no proofreading activity. So when mutations arise at random, It is sort of like when you're writing or when you're typing, you make spelling errors, but you can go back and correct those spelling errors. Viruses have no way of correcting those, especially RNA viruses have no way of correcting those mistakes. And if that mistake happens to help the virus in some way, that mutation propagates and takes over. However, coronaviruses produce this enzyme called exonuclease, and exonuclease has an error correcting function. So, as a result of this, coronaviruses have a low mutation rate of all RNA viruses. Uh, Coming back to the identity of this virus with other known viruses, the closest homology is to some coronaviruses found in bats, especially to two viruses that were discovered in Kunming province in China, eastern China, in 2018. Those viruses show 96% homology to the COV2 virus. And this leads to the thinking that this virus has jumped in the human population from bats. Uh, there is a virus found in pangolins, which also has more than 90% identity. And there has been speculation that the virus first jumped from bats into pangolins and then into humans. But there are other hypotheses saying that it jumped directly from bats into humans, and it has taken some time to evolve in humans and become a pandemic potential virus. The first coronavirus that emerged in 2002 has 80% homology to this virus. The MERS virus, which emerged in 2012, has 55% homology. And the common cold coronaviruses that are already endemic in the human population have only 50% homology. So we have a virus that is really quite unique that's causing this pandemic. Now, this looks at an artist's rendering of the virus, and what you will find is the virus structure is really dominated by one protein, which is called the spike protein. And the spike protein is assembled as trimers on the surface of the virus, or the envelope of the virus. There are two other proteins within the envelope. One is called envelope, the other is called membrane, but those are minor compared to spike. And spike also protrudes from the surface. So it is really the first protein that makes contact with the target cell, binds to the receptor on the cell, and enters cells, which is sort of shown in this cartoon over here. The red knobs are the spikes. These spikes bind to the receptor, which is the ACE2 molecule on the surface of cells, shown here in yellow. And once this binding takes place, then the virus is internalized and the replication starts. So all the efforts towards vaccines are to make antibodies that will disrupt this interaction. So if you develop antibodies that bind very tightly to the spike protein, then these antibodies will prevent binding of the virus to the receptor, and therefore it will be an aborted infection. Now, looking across the genome of the virus, we find that the 5' 2/3 of the genome shown in pink here uh, is made up, codes for the non-structural proteins. The non-structural proteins all shown in blue here, are proteins that have enzymatic functions, proteins such as replicase, methyltransferase, and so on and so forth. We don't really need to go into this. It's just that these proteins are the ones that are required for replication of the viral genome in cells. And part of that is the exonuclease, which, as I said earlier, has an error correcting function. The 3' 2/3 or 1/3 of the genome codes for structural and regulatory proteins. The structural proteins are shown in purple. So you have the major spike protein here, you have the envelope protein, membrane protein, and another protein called the nucleocapsid, which essentially binds to the RNA genome and packages this genome into the virus particle. And shown in green here are regulatory proteins that alter the cellular environment for the virus to be able to replicate well in those cells. The virus replication is fairly typical of a positive strand RNA virus. So the first step that happens is the virus binds to cell surface receptors. And as I showed you earlier, this binding happens through the spike protein on the virus and the ACE2 receptor on cells. After this binding, uh, there is uh, uh, there is cleavage of the spike protein into S1 and S2 by a protease called tempress, which is present on the cell surface. This leads to fusion of the viral membrane and the cellular membrane. Uh, and the virus enters through endosomes that are formed through this fusion. The endosomes get acidified and release the viral genome. Now, there are these two steps that can be interfered with. So, for example, convalescent plasma that carries antibodies those antibodies can bind to the virus and prevent its binding to receptors. Uh, you could also have drugs like chloroquine, hydroxychloroquine, azithromycin, that are basically ion channel inhibitors, proton channel inhibitors that prevent acidification of the endosome and therefore release of the viral genome into the cytosol. But once the viral genome is released, being a positive sense RNA virus, it's like mRNA. The mRNA is translated uh, in first into the non-structural proteins. These proteins, if you recall, have enzymatic function and therefore replicate the positive strand RNA into negative strand RNA and the negative strand back into positive strand. So this is the amplification step of the viral replication. And this step can be inhibited by replicase inhibitors such as remdesivir, favipiravir, and ribavirin, which are essentially nucleotide analogs. Now, when the positive strand RNA is made, you make a genomic RNA and you also make subgenomic RNAs. It is the subgenomic RNAs that make the structural proteins, and then these structural proteins package the full length RNA into new virus particles that get out of cells and start another round of infection. Uh, so that's really, in very simple terms, the replication cycle of the virus. Now, a number of mutations have been observed in the virus. There is a lot of genomic diversity in the virus that is circulating around the world, and I'll deal with that over the next few minutes. So if you look at the evolution rate of the virus, it is fairly low. This 10 to the minus 3 per nucleotide per year translates to about 2 to 3 nucleotides per month. So you might ask why, if the rate is so slow, and by the way, this is about 1,000 times slower than the flu virus, the influenza virus. But if the mutation rate is so slow, how come we have so many variants? How come we have so much genomic diversity? There are over 100 million people infected. So the virus has had a huge amount of opportunity to find substrate and to be able to multiply and amplify in 100 million people. And every day the virus produces hundreds of millions of new particles. So even mutation at a very low rate is going to amplify because of this increased transmission and increased ability to replicate. Now, if you do phylogenetic analysis, if you look at the mutations and the groupings that have come about since the virus was first seen in December 2019 till now, February 2021, you will see two things. One is that the original virus that came out of Wuhan, shown here in dark blue, has now become a minority variant. A new variant started emerging in early 2020, this arm of the tree, and that this arm of the tree is the one that has really expanded over the past year. And within this arm, we are now seeing other variants emerging. So, for example, this deep orange color is the UK variant that you may have heard about. The the slightly lighter orange is the South African variant, and this red one is the Brazil variant that is now emerging. This is another way of showing it, just to show that sequences that are at the periphery of the circle are more recent additions. So whether it's the UK variant or the South African variant or the Brazil variant, these are very recent additions to the variants. Uh, the, uh, the illustration at the bottom essentially shows the fraction of each group that is now circulating in the population. So you will see that the dark blue has is very low, maybe only about 1%. And this is the virus that originally came out of Wuhan, but it has now been taken over by other viruses. And now these orange lineages, the UK variant, the South African lineages, these are now emerging. So that's the current situation of the clades. Now, I'm also showing you the clades that are there in India uh, for one reason. If you look at this figure, and although only about 50 different viruses sequenced from India are on this tree, it illustrates the point that where the virus circulating in India is no different from the virus circulating in rest of the world. There have, people have been saying that, in India the mortality is low simply because we have a weaker virus. There's nothing like a weak virus. It's the same virus that's there everywhere else in the world. So in India, if you see, we have about 11 million confirmed cases. There are roughly 5,200 viral sequences from India that are available in the public database. So that is about a sequencing density of 0.05%. As far as variants of concern are concerned, uh, India has been able to detect 154 UK variants. 123 of those are from travelers. 31 of those are traced to contacts of travelers who came from UK or other countries. By now, we have also detected 5 South African variants, all coming from Maharashtra, and 1 Brazil variant also coming from Maharashtra. At this point, the government of India has set up an interministerial group, which is called the Indian SARS Coronavirus 2 Consortium on Genomics, INSACOG, which is ramping up sequencing. There are 10 sequencing labs that are going to be sequencing at a density of about 5% daily cases. So we have the vision to go from 0.05% to 5%, a 100-fold increase in our sequencing capacity, which will allow us to catch any domestic variants that are emerging before they actually become variants of concern for us. So that's really a very good move right now. This is a paper that was just published from CCMB in the Journal of Biosciences, which reviews all those 5,000 plus sequences that have been done from India. And it gives you a sense of how the virus has emerged, how the clades have emerged in India over time. It also gives you a breakdown of the states and how clades have emerged in different states. What is important to see is that while the frequency of mutations is quite high in the non-structural genes, ORF1A and ORF1B, uh, these may have effects on viral replication, but these are not going to have any effects on vaccines or have any effects on virus entry into cells, simply because these genes are expressed only inside cells and have an enzymatic function. But the spike protein, there are over 750 mutations seen in India in the spike gene, and shown below is the spike protein, different regions of it. And the mutations are really concentrated in either the N-terminal domain. Some of these are deletions that cause PCR tests to miss the diagnosis. But many of these mutations are emerging in the receptor binding domain, which is essentially the domain that binds to the ACE2 receptor, and also binds to neutralizing antibodies. So this could be a matter of concern in the days to come. Now, remember I told you that in January, another mutation arose that has now taken over the world. And it is essentially this mutation, a mutation at 614 position in the spike protein that converted an aspartic acid into a glycine. And this glycine variant shown in yellow, you will see, has really taken over the world since its emergence sometime in January 2020. The original virus still circulates, but circulates to about only 1%. About 99% circulation is there of the G variant. Now, why has the G variant overtaken? So people have done tests. So for example, two different viruses, the D614 virus and the G614 virus, have been taken, and cultured cells have been infected with this to find that the G virus makes higher numbers of viruses, replicates better than the D virus. Now, when you uh, when you also infect something called a human airway model, which is more similar to the lung epithelium in our cells, in our lungs, then the same thing happens as well. So both in 2D cultures as well as sort of 3D mimicking cultures, this has been seen that the virus that carries G at 614 replicates much better. What has also been seen is that the G virus outcompeted the D virus when equal amounts of this virus were taken for infection. So, what we say is that this virus has increased viral fitness, and it produces higher copy numbers. Uh, these viruses have also been tested in a hamster model by inoculating in the nasal cavity of the animal, and again the same result. When you look at nasal washes, you find the G variant to be present in higher concentration than the uh, D variant of the virus, and in direct competition, the red virus outcompetes the blue virus. But thankfully, this mutation has not given the virus any ability to evade antibodies. So, if you try and neutralize with a serum from exposed people, both viruses get neutralized equally effectively. So, there is no concern that this virus will evade antibodies or vaccines. However, since it replicates at much higher number, it infects more readily. The transmission has increased. So the transmission, as transmission increases, more and more people get infected. As more people get infected, uh, the virus has more opportunity to make errors, and some of those errors will get selected. It also puts more people into the severe disease category and more numbers of people in ICUs, although percentages remain the same. Uh, So a paper was published very recently, just last week, from the National Institute for Biomedical Genomics in Kalyani, where they looked at this, what contributes to this, the D to G change at 614 position. And basically what they see is that if you make this mutation D to G, it creates an extra site for a protease called elastase. Elastase is a protease that is produced by neutrophils. It is secreted from neutrophils, and it is present in the lungs. When there is inflammation in the lungs, neutrophils get to the site of inflammation, and this protease is secreted there. This protease is inhibited by uh, by a protein called alpha-1 antitrypsin inhibitor, or AAT. And AAT frequency, if you see in human population, uh, AAT deficiency is quite prevalent in Europe and North America compared to East Asia. The data from India is not available in this paper, but if we are presuming that we are more close to East Asians than we are to Europeans and North Americans in our genetic polymorphisms. So what does it really mean? So here is the hypothesis that the NIBMG group has proposed. If you look at the left part of the lung, so here are neutrophils that are in the lung. They secrete this enzyme elastase, but in the lung you have alpha-1 antitrypsin, and it inhibits the elastase, not allowing it to clip the virus, and therefore it is normal viral entry, and in presence of normal levels of alpha-1 antitrypsin. But what if somebody has alpha-1 antitrypsin deficiency? What happens then is that the protease that is secreted by the neutrophils is not neutralized because you have less amount of alpha-1 antitrypsin. And because of that, this protease clips the S protein of the virus into S1, S2, enabling more virus to get inside cells. And this causes enhanced viral entry, enhanced inflammation and tissue damage, leading to more severe disease. So this is a hypothesis that really explains why the disease has been symptomatic disease, and mortality has been less in East Asians compared to in Europe and North America. An attractive hypothesis. One way to really support this is to measure alpha-1 antitrypsin levels in lung infiltrates from people from various regions of the world to be able to support this hypothesis, but certainly a very interesting hypothesis. You may have heard a lot about these viral lineages. So there is one lineage called the UK variant, or the B117 lineage, which has now been discovered in more than 80 countries. And a lineage is something that brings multiple mutations together in the same virus. So, for example, the UK variant lineage has 23 mutations, out of which 17 mutations are non-synonymous, means that they also change an amino acid at that site. And out of those 17, 8 non-synonymous mutations are in the spike gene. And this causes the increased transmission. This also causes some evasion of antibody response. The other variant of concern that is circulating is a South African variant, which has now been reported from more than 40 countries. And this also has some very key mutations in this spike protein that have come together in this lineage. There are other lineages that are developing. So, for example, the P1 lineage is the Brazilian lineage, and there are two other lineages, the 23.1 lineage and the 525 lineage, which are found in parts of Africa, UK, and US. Okay, so how about COVID-19 vaccines? So let us look at the vaccines in development. So if that's a cartoon for the virus, one type of vaccine that is under development is a whole virus vaccine. So in the inactivated vaccine, you can grow up large amounts of virus, inactivate it with a chemical, and use it as a vaccine. So this is essentially what the Covaxin or the Bharat Biotech vaccine is. It's an inactivated viral vaccine. There are really no live attenuated vaccines for COVID that are in late clinical trials at this point, but there are some under development. The DNA vaccine and the RNA vaccine, especially the RNA vaccine, these genetic vaccines have really captured our imagination, because you can take simply nucleic acid sequences, put them into cells, and the cell will convert the RNA to proteins. You can move one step back, the DNA will make RNA and the RNA will make protein. And the RNA vaccines have shown very high levels of efficacy, and it's a completely new platform. The next type of vaccines are these viral vector vaccines, which also take a piece of DNA from which codes for the spike protein and uses a virus to send this into cells. And the most common virus is the adenovirus or the common cold virus. The Oxford AstraZeneca vaccine, which is made by Serum Institute, called Covishield, is based on this principle and uses an adenovirus from chimpanzees. The Russian vaccine and some Chinese vaccines use human adenoviruses. The Johnson and Johnson vaccine that is making a lot of news as a single shot vaccine in the last few days is also a human adenovirus-based vaccine. And finally, you have the traditional viral protein vaccines. So the Novavax vaccine, which has shown very good results, is based on a virus-like particle. So it's a particle made up of protein, but doesn't contain any genetic material in it. So you see that we really have a bouquet of vaccines that are both in development and available to us now. So at this time, there are 4 approved vaccines, 6 other vaccines that have received limited approval, and over 65 different vaccines that are in multiple phases of human clinical trials. And here is a list of those, all those vaccines. The 2 vaccines available to us in India are the Bharat Biotech vaccine and the Oxford AstraZeneca vaccine made by Serum Institute. So that's it's called Covishield. The Bharat Biotech vaccine is called Covaxin. So the question is, would current vaccines work against variant viruses? And there are a number of reports that have come up in the last, basically last 6 weeks, that have tried to look at this problem. Without going into the details of this, let me just summarize this. So here in this cartoon, you see the spike protein in gray over here, and in green is the neutralizing antibody, which binds to the receptor binding domain of the spike protein. Now you notice that the receptor binding domain has some key mutations that are coming up. So for example, the N501Y mutation, the E484K mutation, the 417N mutation, and there's another mutation, the N440 mutation, which is coming up lately, which is a mutation of concern. So basically, this paper published on 17th of February in New England Journal of Medicine summarizes it quite nicely that as of now, viruses which have the full set of South African lineage variations or mutations uh, have a high chance of escaping vaccine-mediated antibody responses. And this is also shown from the clinical trials that were done in various parts of the world. If you look at the results of that trial, the vaccine had 90% efficacy in US, but only 49% efficacy in South Africa. And it was tested at a time when these variants were circulating. So variants are emerging that can be a cause for problem in future, and we need to remain vigilant. So in India, these are the mutations that have come about, and I've shown you the amino acids to really refresh your first year biochemistry. The mutation of concern is this E484K mutation. Why is this a mutation of concern? Because it converts a negatively charged site into a positively charged site at the interface where the antibody binds to the spike protein. This is likely to cause a major disruption in binding. Uh, this is also a very common mutant that has emerged across the world and in India also. And more recently, a variant uh, carrying the E484Q to E484Q mutation has been seen from parts of Maharashtra. So some mutations of concern are coming up in India, but so far only single mutations are being seen in India. These mutations have not come together in any organized lineage. Variants of concern are present in the country. I showed you earlier how UK variants are present in the country, but the community transmission of these variants hasn't really been reported yet. Uh, if you recall, the UK variants are seen in 123 travelers and 31 other people who have been in touch with these travelers. But so far, this has not been seen in people who have not been in touch with any travelers. So that's why we say no community transmission yet, but it doesn't mean it won't come. If we're not careful, it will come. And therefore, genomic surveillance is really needed, and it is needed at reasonable density. And that's why the interministerial group that is now set up to sequence viruses at 5% density is a very welcome move. And this is going to let us find variants before they become variants of concern. I will end simply by asking, do masks help, and should we wear masks even after vaccination? And since we are all scientists and nothing convinces us more than data, here is the data for you. This is a study of 200 different countries, mortality rate in 200 different countries by their habit of wearing masks. So if there are countries that implemented mask wearing within 15 days of first case detection, they have very low mortality shown by the blue line. If there are countries that have mask wearing between 16 to 30 days of first case detection, they have slightly more but controlled mortality. However, countries that have not implemented masking, even after 60 days of first case detection, look at where the mortality rates are. So this is data from 200 countries. You can't really deny it. How else do masks help? Now, here is an experiment which was really done for bacteria, but you remember that viruses are also present in respiratory droplets, and therefore they transmit very similar to how bacteria transmit. So here is an experiment where you sneeze, sing, talk, or cough on a culture plate without wearing a mask and with a mask, and you see the difference. So masks help, and we must continue to wear masks. What masks do is reduce transmission, and if they reduce transmission, they will reduce viral mutations and reduce the development of variants of concern. That's a plain and simple fact. Uh, Everyone in India is now thinking that the outbreak is over for us. We have low numbers of cases. Two vaccines are available. No need to wear masks. Not true. While vaccines are available, would a common man, would you and I get the vaccine now? The answer is no. Look at the vaccination rate in India uh, till last week. Uh, we have started out by giving roughly 18,000 roughly 1.8 lakh vaccinations a day, and have gone up to about 7 lakh vaccinations a day now. This is really good progress, but look at how much time it will take to vaccinate the population with 2 doses. If we want a 20% coverage of the population, which is essentially what the government wants in the first 7 to 8 months, 300 million people, 30 crore people that the government is now targeting is roughly 20% of India. If you were able to give 20 lakh doses per day, and remember, we're not there yet. We are only about 7 to 10 lakhs at this point. If you are able to give 20 lakh per day, then it will take us 9 months to vaccinate 20% of the population. So this is really a sobering figure. Don't think that you're getting the vaccine tomorrow. You're not. And that's why continue to wear a mask. That's the only way to protect yourself and to protect others. Thank you very much. I'm done. And if there are any questions, I'll be happy to take.
Thank you, Shahid. It was wonderful. COVID-19 in global perspective. Okay, so we don't have too much time left. Maybe one or two burning questions, if somebody has, please ask.
Vijay, may I ask a question? This
Please
is
go ahead. Please go ahead.
Professor Jameel, this is Vijay Subramanian. That was a really lovely talk. Thank you so much for all that information. I have a quick question. So you talked about mutations on the spike protein. What do we think about mutations in the exonuclease proofreading activity? Because if that happens, then the virus is going to mutate really, really fast, right?
Mutation in the proofreading activity, you said?
Exonuclease proofreading activity, because right now it is giving us 1 or 2 mutations per month, like you said.
Right, right. No, interesting thought. Yes, if the proofreading activity is compromised, then you'll have more mutations. But also remember one fact, that most mutations are bad for the virus because they interfere with, most mutations are either neutral, meaning that they don't affect function, or they are deleterious to the virus. There are only some mutations, a small percent of mutations, that give some added advantage to the virus. The ones that are deleterious to the virus, we never see those mutations because those viruses disappear from circulation. So yes, if the rate of mutation increases, certainly is going to be a cause for concern. And that's why it's so important for us to limit transmission. And that's why I keep saying the only way to limit transmission is to wear masks. And I'm so happy to see all of you around the table wearing masks.
Thank you. Just one quick follow-up. So when we are sequencing, when you mentioned India is sequencing the virus, are we sequencing the entire virus or just
yes,
the spike?
Yes. No,
Thank you.
Entire virus. Yeah. It's a huge effort going on right now. Yeah.
Sir, you're muted, Professor Vijay.
One clarification, Shahid. This is about the epidemiological fact where we are still about 20-22% seropositive at a populational level. Does that mean clearly we are sub-threshold in terms of so-called herd immunity?
Absolutely.
Okay.
Yeah, but remember that 22% figure did not include some major cities. So if we consider that, then cities have much more spread of the virus, and cities may be closer to this idea of herd immunity, but we don't really know that. Firstly, we don't know what percent is required for herd immunity. Herd immunity is a concept used more for vaccination than for infection. So it would really be a mistake to depend upon herd immunity.
Okay.
May I ask a quick question, Vijay?
Please go ahead.
Yeah. Shahid, this is Ram Kumar, a developmental biologist at ICER. What underlies the pandemic potential? Is it only the mode of transmission? And also, what is the basis for survival for it to be airborne?
Basis for survival of
the
the virus?
capacity, survival of the virus. For it to be airborne, I imagine it's the survival that matters.
Yeah, so the virus is definitely airborne because we throw it out when we cough, when we sneeze, when we talk. But one fact to remember is that the virus is present in droplets. And the bigger the droplet, more virus is present in the droplet, right? But bigger the droplet, heavier is the droplet. So it will fall very close to the source. Only the very small droplets, which also have small amounts of virus, are going to distribute. So if you're in closed spaces, especially if you're in closed spaces in such an airflow that somebody coughs upstream and you are downstream of that airflow, you have a higher chance of getting it. But if the one who's coughing is wearing a mask, he or she is putting out much less virus, and you downstream of that airflow also are wearing a mask, that cuts down transmission to less than 5%. If both are wearing a mask, it cuts down transmission to less than 5%. This is really a proven fact. No matter what people tell you, don't believe that masking is important.
Thank you, Shahid. We really have to move on to the next talk now.
Yes. Thank you very much.
To all the audience, if you have more questions, please shoot directly to Shahid Jameel. He is email accessible, and I'm sure Shahid will be more than pleased to respond by email replies. Thank you very much.
Thank you.
So we will now move on to the next talk by Ramji Raghavan sir, and the topic is very exciting: how to build a creative and innovative India. All of us are interested in hearing this talk. Thank you very much, sir. Thank you. Please go ahead.
Thank you, Basutkar. It's a pleasure and honor to be here today. Let me start with a story. In 1884, a 5-year-old boy was lying ill in bed when his father brought him a gift to cheer him up. And like any young child, the boy was intrigued, got up with a smile, opened the gift and started playing with it. And it was a magnetic compass. And whichever way he held it, it always seemed to point in one direction, the magnetic north. Now, we all get gifts, and we all react differently to gifts. But in this particular case, the gift turned out to be a momentous gift. In his 70s, Albert Einstein, who was that boy, said that it was the magnetic compass that made him wonder if there was an invisible force behind everything in the universe. And as we all know, he spent his life trying to find that, and in the process, revolutionized science and human thinking. Now let's fast forward 124 years later, in 2008, in a place called Kuppam, which is about 100 kilometers from ICER Tirupati, on a hot summer's day, 2 village girls, Rani and Roja, went and sat under a tree to escape the sun. Sensible thing to do in the summer. And they began talking, and Rani asked Roja, Roja, do you ever wonder why you feel cool or cooler sitting in the shade of a tree? And Roja thought for a second and said, maybe it's the leaves and branches of the tree that shield us from the sun. And the girls continued talking until the aha question popped out. Would different leaves have different cooling effects? And that question led to a project, and working with teachers from the Agastya Foundation, 8 months later, the girls won a prestigious Intel Iris National Science Award, competing with the best and brightest kids from across India. Amazing story. So what do these two stories, at one end, a young Albert Einstein, and another two girls from Kuppam have in common? Well, I believe it's the transformative value of curiosity, the magic of wonder, and the power of passion. In fact, Einstein attributed his great discoveries to three factors: curiosity, obsession, and dogged endurance. You know, to the question, what is the world's most vital resource? My answer would be curiosity. It's the most vital natural resource. In fact, a friend of mine at Stanford said it very well. He said, the biggest city in the world is not London or New York or Mumbai. It's curiosity. Curiosity with controlled inquiry can lead to creativity, which involves observation, discovery, experimentation, association, and all the rest of it. Creativity in turn leads to new ideas. Ideas can lead to invention, and inventions through inventiveness can lead to innovation. Innovation increases productivity, and increase in productivity leads to an increase in prosperity. So that's a, if you like, a virtuous cycle from curiosity to prosperity. In fact, if you look at the golden age of India, you know, or North India to be more specific, during the Gupta period, there were three reasons, they say, for the golden age. One was economics. They had these guilds that were generating money. So you need money to invest, to spend. The second was enlightened rulers. The rulers patronized the arts. They patronized the sciences of the time. They encouraged learning. And the third was curiosity. Much more recently, the Nobel laureate Ned Phelps has written a book called Mass Flourishing. And he concludes that the reason Britain was so prosperous and dominant in the 19th century, and America in the 20th century, at least up to the 1970s, was because of what he called indigenous grassroots innovation. Innovation that came ground up, right? So he likened the economy to a sort of imaginarium that encouraged discovery, experimentation, and so on at the grassroots, which led to innovation, which caused increases in prosperity, in productivity, and therefore prosperity. So curiosity is really fundamental to creating a prosperous nation. And yet, in India, we seem to have killed curiosity. Maybe it's not just India, many other parts of the world as well. You know, in the US, they did a study and they found in schools, and they found that 98% of children between the age of 2 and 4 ask questions. Something we can all relate to, or our parents can relate to. By the time the children hit middle school, that has dropped from 98% to 54%. And by the time they leave high school, it's dropped to 25%. So you start school as a question mark and end up as a full stop. It's very sad. Now in India, I visited hundreds of schools, elite schools, as well as village schools all over the country. And while in America, they might say that 25% of children ask questions by the time they graduate from high school, I've seen in India, it's probably in the low single digits, if that. So my submission is, if you want to build a creative country, an innovative country, that has to change at the grassroots. And it's with that in mind that in 1999, my father, K.V. Raghavan, the former chairman of the Atomic Energy Commission, P.K. Iyengar, the former principal of Rishi Valley, Dr. Balasundram, and a colleague from Rishi Valley, Mahavir, started Agastya Foundation. And we actually had a very interesting brainstorming right here in Bangalore at the ITPL. And Iyengar said, look, Ramji, your idea of creativity is very interesting, because I wanted to nurture creativity in a new school. Why don't we probe that more deeply to figure out what is it? So we began to ask a lot of questions. What makes people creative, innovative, great problem solvers? Is it something in their genes? Are they born like that? We're all born curious, but are we born creative? Or is it a skill that we can learn over time? And we said, look, the creative process involves observation, discovery, experimentation, association, and application. And the foundation of all of that is curiosity. So my question then was, can I learn to be more creative? And Iyengar said, look, you're a non-science guy. I will give you 100 counterintuitive science experiments, simple science experiments. And I guarantee you, in a couple of months, you will begin to see the world in a very different way. You will begin to ask more questions, be a lot more observant, and so on. So, while it may not be possible to be a Ramanujan or an Einstein, it's certainly possible to raise the speed limit of creativity, if you like, for any individual. So if you, Ramji, are at 0.5 or 2, to put it crudely, through certain interventions, we can certainly raise your creative output and potential to a 4 or 5. And if we did that at scale, we'd bring about transformation. So we said, let that be the mission of the Agastya Foundation, to spark curiosity, nurture creativity, and importantly, instill confidence and caring. And confidence and self-efficacy is so important. I remember years ago going to a village to find out if we were having any impact near our campus, which is in Kuppam, that Ganesh has very kindly visited. And a teacher pointed me to a girl, Uma, and said, why don't you go and ask her? So I asked Uma, you've been coming to our campus for several months. Any impact, Uma? And I thought she'd say, I'm doing very well in my science subjects. But she didn't say that. She looked at me and said, sir, I'm not afraid to speak anymore. And that was sort of an aha moment for me there and here. And I thought, wow, one of the effects of our interventions seems to be to raise the self-belief of children. And that is actually central, because you can be very curious, but not particularly confident. You might even be creative or have that potential, but you might not believe you can do something or achieve something. So that sense of confidence and self-efficacy, we said, is so important. So that became the foundation's mission. And then we asked the question, great, how are we going to spread this? I had a vision of starting a school, a sort of Taj Mahal of creativity, which, by the way, we have built in Kuppam, and I would invite all of you to visit. And if you can't, then maybe see it on the internet. I'm going to be showing a film later today in my talk that will show you glimpses of the campus. So I thought, let's build this Taj Mahal of creativity. And then the question was, how would we do it? And we said, how about hands-on experiential learning? Now, I'm an MBA, as Basutkar pointed out in his introduction. And while I studied science at school at Rishi Valley, I was never deeply into the subject. So I'm a non-science person in that sense. But I was surrounded by scientists and engineers and so on. And they said, how about hands-on science education as the medium to spark all these important creative traits? So and why hands-on? Because the education system was so focused on chalk and talk. I talk, you listen. Exam-oriented. Kids couldn't relate to what they were learning. They couldn't apply it to real life. They'd actually not seen something, and experimented. So those sensibilities were completely dormant. So we said, let's do hands-on experiential learning. The other thing we found was that cognitive scientists told us in the literature that The human brain on average retains 5% of a lecture in its long-term memory. 50% of what you see and hear, 70% of what you discuss with someone, 80% of what you personally experience, good or bad, and over 90% of what you teach to others. So we said, look, if you look at it like a pyramid or triangle, the education system is focused on chalk and talk, literacy and all that, and literacy is very important. But our unique proposition should be to offer hands-on experiential learning. So that became the strategy. And then the next question was, what should we do? And I said, let's go and get some land to create a campus. And we got the land, barren wasteland, rocky wasteland in Kuppam. That was great news, except we ran out of money and we didn't have money to build buildings to create this Taj Mahal of creativity. So what do? I quit my job as a banker in London. A lot of people were putting in time pro bono. So we needed to do something. We came up with the idea of a mobile science van. And the Homi Bhabha Center for Science Education gifted us a trunk full of low-cost, counterintuitive science experiments. And one of our people, a former tractor driver, became the first teacher, took these experiments in the van into the villages near Kuppam. And a few weeks later, he came back and told us it's a super hit. No one's ever experienced anything like this. So we said, well, keep doing it till we come up with a better idea or more money. But it also provoked us to start thinking, is it possible to raise the speed limit of creativity of an entire nation? And if it is, can it be done by a single campus? And the answer was no. We need to find ways of scaling our ideas across the country. And how would we do that? And the answer came to us in the form of this little top. I hope you can see it. It's called the tippy top. So if you put it on a surface, it sits on its bottom, and I don't need to explain to ISA students and faculty why that happens, right? But if you spin it, almost miraculously, it tips over and begins to spin on its stem. Now, this is a toy made in Chennapatna that costs all of 10 rupees. But the beauty about it is really a symbol for transforming a nation. Because when you see it tip over, you go, ah. And the ah is so important because there are millions of people across the country whose minds have gone to sleep because of lack of stimulation, mind-numbing education. They've lost interest. And when you do something like this and you see something unexpected, you go, ah. And that begins to awaken your mind and stimulate your senses. And when that happens, you might wonder, you start playing with it, and you might ask the question, why does that happen? Now, I have a friend at Stanford, Tadashi Tokieda, who studies the science of toys. That's all he does. And he looks at counterintuitive things, okay? Now, I'm not a scientist, but I've often wondered why that happens, gone to the internet. And in any case, if you do find out, in fact, there's a picture on the internet of Wolfgang Pauli and Niels Bohr in 1951, playing with the tippy top. And you can see them in their suits with a smile, probably wondering why that happens. And maybe they figured it out, mentally on the spot. But I've never really understood it. But certainly I've enjoyed it. So if you do understand it, you experience the famous aha effect. So what we said is curiosity, aha is the process of creative thinking and comprehension, are two profound ways of learning. But there's another very important element, and that is the sense of play. This never ceases to give me a sense of joy when I play with it. I carry it with me wherever I go, or I used to go in the world. And it also makes a very nice gift, especially to children, and a very low-cost one. And the third element is joy of learning, which is the ha-ha effect. So we said, look, Agastya's mission, worded differently, more simply, is to disseminate the spirit of aha, and ha ha. You know, the famous Romanian sculptor Brancusi famously said, simplicity is complexity resolved. So our mission was boiled down to 3 sounds or interjections: ah, aha, and ha ha. Now, what happens when you experience that, whether it's a tippy top or a piece of paper or any other science experiment or even art activity or activity in the humanities, it begins to change your thinking. It creates what we call inciting moments. You know, Einstein experienced, in his own words, an inciting moment. So did Rani, right? I'll give you another story. There was a girl called Maunika who came to talk to us in her early 20s a few years ago. And she was one of those children. Basudkar was talking about peer-to-peer learning. We have a program where we teach children to teach children, and she was one of those. Her story is fascinating. She had no interest in academics and studies, and her grades in school were absolutely bottom. So her father decided that he would get her married off as soon as possible, which she found mortifying. She didn't want to get married. But the father said, you're absolutely hopeless in school. You're better off getting married to someone. And as luck would have it, a friend of hers asked her if she'd been to the Agastya campus. She said no. She said, well, come along with me. So she brought her. And again, as luck would have it, the teacher at the Agastya campus looked at Maunika and said, Maunika, show me your finger. And Maunika showed him her finger, not knowing why he asked her to do that. And he quickly pricked it, and she literally went, ah, because she felt the pain. Took a drop of blood and told her a while later what her blood group was. Now, when Maunika was talking to us, she said, up until that point, I had absolutely no idea that there was such a thing as a blood group. And suddenly I realized, it was an epiphany, that there was a world of knowledge out there that's fascinating, that I need to be a part of. Guess what happened? She began to take interest in her studies. Her grades began to go up. So she went to her father and said, look, I'm doing well at school. Can you postpone my marriage? And he said, no, not really. So she said, look, if I come first in school, will you agree to send me to college? And then I get married after college. And he didn't believe she could come first in school. So he said, fine. And guess what? She came first in school. And the man was an honorable person. And he said, okay. And she ended up at Tirupati, SV University. I remember as a schoolboy at Rishi Valley playing cricket matches against the university and always losing. And there she became a student leader of 3,000 students. Graduated with a BSc or BSc Tech degree and came and spoke to us about how that pinprick moment transformed her life. So what we said is, we should really be scaling these pinprick moments, these inciting moments, through low-cost, hands-on experiential methods. And we started to do that. And over the years, we reached over 15 million children and 250,000 school teachers. School teachers, because it's so important to create that force multiplier effect. So that has been our strategy to really create this bottom up, right? Curiosity fueled innovation, an innovative mindset, a creative mindset that can transform this country. Indigenous grassroots innovation, if you like. And then we set up an innovation hub along with the government of India, and that is focusing more specifically on how to find solutions for problems. This girl in Kuppam, you know, whenever I used to take the road to Kuppam, as I neared Kuppam, I would hear the sound tick, tick, tick. And you look out the window and you see these guys chipping away at the granite boulders. Well, one of these girls who came to our innovation hub said that her father would come home every evening with his hands bleeding. It's very sad. So she worked with her friends and created a glove that he could use that was reusable, that would prevent his hands from bleeding. So things like that, finding solutions to your own family's problems or your community's problems is something that we are promoting, and we intend to do that at scale. Today, Agastya Foundation has about 200 mobile science vans, 100 labs on motorbikes, about 100 science centers, and in about 400 or 500 villages, we run a night village school program called Operation Vasanta. And pre-COVID, we were reaching about 1 million children every year directly face-to-face, and another 1 million through the teachers we trained. And then, of course, COVID came and brought the whole world to a standstill, as it did Agastya. And fortunately, we had and continue to have this culture of ceaseless innovation. You know, it's about practicing what you preach. If you're going to spark curiosity in a child or a teacher or nurture creativity, you need to ask yourself, are you yourself curious? Have you done anything creative or innovative? So in Agastya, we've also looked at ourselves inwardly and said, are we an innovative organization? And what do we need to do to build an innovative organization? And over the years, we created this momentum of ceaseless innovation. You know, it's like a permanent revolution. And people would say, hey, you know, maybe we're overdoing it, too many projects, too much attraction to novelty and so on. But when COVID hit us, these qualities came to the fore and saved us. Because very quickly, we applied our skills in innovation and curiosity to pivoting to online digital education. And very quickly, the team came up with Explore, Play, Learn modules. And then we said, but how do we maintain our focus on hands-on experiential learning? Because a lot of online digital is not hands-on in that sense. So we said, how about if we transform the child's home into a laboratory? So let's teach the child how they can create experiments from everyday available materials at home. And that became a kind of new blended learning model. And we said, hey, COVID has been awful. The schools have closed down. But you know what? We have created this new blended learning model. And a couple of years down the road, the real opportunity is, while we might be one of the largest players in the world in terms of hands-on, face-to-face learning, if we combine it with online digital, we'll become a unique player in blended learning at scale. So that's the new opportunity. And a couple of years ago, I was talking about transforming Agastya, the organization, into Agastya, the movement. And I used to rack my brains on how are we going to achieve it, because you have to think and act very differently. And with COVID coming, it really accelerated our move towards the movement. And digital and online is virtual learning is a very important part of that. So we are in the midst of rejigging the foundation, and we have set an extremely ambitious goal. We reached 15 million children in 20 years. And now we're saying, can we reach 100 million children in the next 5 or 6 years? So that's a sort of exponential trajectory. So the whole organization is alive and abuzz, and I suspect even a little nervous about how we're going to do that. But I have no doubt we'll do it, because while our mission is to spark curiosity, nurture creativity, instill confidence and caring, what we call the C4 gene, inject that into the education system, the larger purpose is really our dream of a golden age for the country. And to achieve that, while we have reached a lot, we've got to have far more impact. Now, the good news is, you know, when we started Agastya 20 years ago, there were very few people outside the scientific community who understood even what curiosity meant. I mean, I was one of those. I worked for a top financial institution, but I can't say that I really looked at things like curiosity, creativity in any depth. So we were pioneers to a large extent, along with a few others. 20 years on, it's music to my ears and to all my colleagues' ears to hear about a fundamental shift that is being advocated by the new national education policy. As the Prime Minister said, it represents a shift from what to think to how to think. Now, at the policy level, that is fantastic. And we are committed to working with the state governments to make it happen.
We apologize for the loss in connection, and we hope Shri Ramji will be back in a minute.
Yeah.
Is some problem in his place, or is it something connected with our connectivity?
I think there, I'm trying to reach him.
Oh, it's there. Maybe you can use mobile and check with him.
I'm calling you.
Yeah.
So then we will realize.
Right.
Yeah.
I guess he's trying to rejoin. So he responded, he's trying to rejoin because he's going on with the talk. So he did not realize that he was offline. Yeah.
Okay.
He's rejoining and it may take a minute.
Convey the message.
Yeah, I've done that. Yeah, he's rejoining, Vasudha.
Yeah, I think.
So you're muted? Yeah. You're still muted.
Sir, you are muted. Please unmute.
Yeah, can you hear me now?
Very
Yes,
clearly.
yes.
Okay, I'm sorry. So where did I stop? At Rani?
We were enthralled by you, so we don't know where you stopped.
Okay. Okay. So, some of you might ask me, might wonder what happened to Rani. And I had forgotten about her. And then in the last quarter of 2019, I think it was, a colleague of mine came to me and said,
yeah, so
can you hear me?
You just started about the education policy and then we moved on from there. That's
Okay.
when.
So did I mention the shift from what to think to how to think?
Yes, you did. Yeah.
No.
Okay.
Okay, we lost it then.
Okay. So fundamentally, we have spent 20 years doing this. And it was, sometimes you do things in life and you might not see the rewards of it or the impact of it in your lifetime. And that's okay, because that's the nature of things. But it was music to my ears when the new national education policy was announced. And even the Prime Minister said that what it, at its essence, represents is a shift from what to think to how to think. And that is precisely what Agastya had pioneered, and others as well in this country. But we did this at scale. To see the acceptance of that philosophy at a national level, I think is a major, major inflection point. Now, you might say, that's great, but the real proof of the pudding is in the execution. And I agree with you. And Agastya is committed to working with state governments to make that happen, where the rubber meets the road. And even if we achieve a fraction of what the country would like to achieve, I think it'll be a big step forward. It might well be the beginning of a new golden age for the country. Because now what we are saying is what Iyengar told me 20 years ago: focus on curiosity. That will solve half our problems, right? In fact, one of our biggest donors, Rakesh Jhunjhunwala, the stock market investor, when I went to see him in Bombay, he said, I will commit to giving Agastya 50 crores over 10 years. And I was quite taken aback. And some years later, he told me why he decided to do that, because he said, you're the first guy who came to me and said, I'm not starting an education NGO. I'm interested in sparking curiosity. And he said, I am what I am because my father allowed me to be curious, right? So it's so important for the vital health of this country, curiosity. And the NEP, I hope, represents a major change in the educational direction of this entire country. Now, some of you might be wondering what happened to Rani. And often I wonder what happened to these kids as they grow up. And we have a process where we try to track their progress. Well, in the sort of third quarter of 2019, a colleague of mine said, Rani wants to see you. And I said, Rani of the Intel Iris Prize? And he said, yes. So Rani came to see me. And I said, so Rani, how are you? And what are you up to? And she said, I would like to take a photograph with you. So I said, first, tell me what you're up to. And she said, I've just been admitted to a PhD program in a university in North Karnataka in the life sciences. And I was thrilled. This is the daughter of a marginal farmer. And I said, Rani, it would be my honor to have my picture taken with you. You're the VIP. That was the sort of transformation. Okay. So having said that, I would now request Anuradha, we have a short film because, the truth is more easily appreciated when seen than heard. And if Anuradha could put up the film, I will conclude with my address and be delighted to answer any questions.
Yes, yes, you can start.
First, ideation.
We're just starting soon.
Focus. Ideate. Innovate. Enable. The Changemakers Season 2 in association with CNBC TV 18.
Hello and welcome to Season 2 of The Changemakers on CNBC TV 18. I'm Ritu Bhandari. In this series, we showcase how India Inc. and the social sector, through their groundbreaking corporate social responsibility initiatives, are creating a positive social change. Organizations that prioritize the health of the planet, whose actions are people-centric, that eventually result in better economic opportunities for everyone, are today seen as the change makers. Well, there is no second opinion about the crucial role that education plays in shaping an individual's personality. Even though India's literacy rate has registered some growth over the last few years, it has not been able to impart the kind of education that modern times demand. The new Education Policy 2020 emphasizes on this much needed shift from what to think to how to think. Several studies on the state of education in schools have also shown that the curiosity levels among children decrease significantly as they progress from one grade to another. Our first change maker, Agastya International Foundation, is changing this phenomenon among children and teachers in India through its various programs. Let's find out how.
Established in 1999, Agastya International Foundation aims to spark curiosity, nurture creativity, and instill confidence in economically disadvantaged children and government school teachers by bringing imaginative and innovative hands-on science education, project-based and peer-to-peer learning to schools, towns, and villages across India. Their mission is based on three simple words: ah, aha, and ha-ha, where A factor stands for sparking and retaining curiosity amongst children. AHA stands for nurturing creativity, and HAHA refers to their commitment to ensuring that they enjoy the process of learning and gain confidence in their abilities. Nestled in the confluence of Karnataka, Andhra Pradesh and Tamil Nadu lies Agastya International Foundation's Campus Creativity Lab, situated near Gudivanka, Andhra Pradesh. Spread over sprawling 172-acre campus, it has been passionately designed and developed with large models to delve into ecology, to learn from and explore.
అగస్త్యలో చాలా అందంగా ఉంటుంది. అగస్త్యలో మనం నేర్చుకోవడానికి చాలా ఫెసిలిటీస్ ఉంటాయి. అక్కడ
Agastya is so beautiful. There are so many facilities to learn. We can learn a lot here as there are so many things to understand. Also, Agastya's teacher teaches lessons which is easy to understand without any difficulty and are so practically demonstrated and yet well understood.
అర్థవంతంగా ఉంటుంది. మా టీచర్లు ఆల్రెడీ చెప్పారు, కానీ దాన్ని వాళ్ళు ప్రాక్టికల్గా చేసి చూపిస్తారు. కాబట్టి మేము ఇంకా బాగా అర్థం చేసుకోవచ్చు.
I know about Agastya since 8 years nearly. So the campus is very pleasant really. The students also, they enjoy very well when they go there. It is really a gift to the rural children. They explain the things and concepts in a practical way, and they show the models, and they are very friendly to the students. Even the children also, they enjoy very well. So we approach the students in the form of curiosity, and we have to creativity. Finally, they have to inculcate confidence on the students by learning that concept. If they learn practically, they never forget the concept.
Agastya's pedagogical framework has incorporated activity-based and project-based learning to hone 21st century skills among children, based on the motto of when children cannot go to Agastya, Agastya will go to them. Their mobile labs and labs on bikes take science, maths, art, digital literacy and design thinking to children in the remotest corner of the country.
Agastya main programs, I would say, are the mobile science lab, which reaches close to 1 million children every year in the remotest corners of India. The Young Instructor Leader program, which builds children as peer-to-peer learners, team builders, and builds character in them in terms of caring. The teacher training program, which helps us to replicate and have a force multiplier effect, because each transformed teacher can transform hundreds of other children into the new way of learning. besides this, we have several programs centered on innovation, design thinking, lab in a box, lab on a bike, etc. We were thinking about how to scale as well as have quality in the program that we have, which is the mobile lab program. And lab in a box was born, where we created 10 boxes and rotated them around 10 schools in 10 weeks, so that each of the 10 schools will have a complete understanding of hands-on learning, experiential learning for all their children through the syllabus that they normally go through. So this has been an outstanding experience. We continuously innovate on our products and services. And as we speak, we are stepping out on a major digital, phygital, and fidgitual kind of programs.
Knowledgeable, enthusiastic Agastya teachers accompany each van to supplement school education and enhance the learning experience.
We have science centers, hundreds of them, and then we have 200 plus mobile labs, about 100 lab on a bike. All of these are manned by teachers, and they go to the remotest corners by traveling 20-30 kilometers every day. They are locally recruited. They relate very well to the children. The children look at them as friends rather than teachers. And in a 2.5-hour session, we are able to catalyze the children to thinking learning is the future.
We are working on extending the physical center of excellences by creating virtual as well as robotic centers of excellences. And in this Agastya 2.0 mission, which entails taking this to millions of students all around the world, we are helping them break the space-time barrier and provide this type of education to anyone, anywhere, anytime. And we wish them all the best.
Agastya is using the blended learning approach of both physical and digital modules to adapt to their changing world that is moving online. More than 12 million children and 5 lakh teachers have been impacted through Agastya's program so far. Agastya 2.0, the ambitious 5-year plan, aims on reaching 100 million children and 1 million teachers by 2025 by adopting new pedagogies, teaching methods, and platforms for learning.
On that note, it's time for a very short break right now. On the other side, we have an organization that is revolutionizing India's workforce by enabling.
Thank you, Anuradha. So, Basutkar, over to you. If there are any questions, I'd be happy to answer them.
Thank you, sir. Thank you very much. It was really enthralling. I am sure my audience will have some questions for you. We leave the session open for questions now. I can start with a question if you don't mind.
Sure.
So we have IITs, ICERs, etc. in our system, and they are what they are. You have Agastya Foundation model. Is it possible to blend both and make it viable?
I think that's a brilliant question. And the answer to you is absolutely. Absolutely. In fact, that might well be the great opportunity to, I know the IITs and ICERs have a great name and reputation. They produce outstanding graduates. But if we can inject these institutions with that spirit of play, curiosity, creativity at an intense level, the hands-on experiential, my sense is they will then become truly global leaders. That's the great opportunity. I would love to explore that. In fact, one of our trustees, Dr. Atre, who used to be the head of the DRDO, he keeps telling me this kind of learning, the philosophy of learning, is as essential in higher education, in universities and colleges, as it is at the school level. So we need to find a way of building bridges to institutions like the ICERs. You know, you are involved in this gifted children program that we are working on, ICER. And already I can see the synergy that where you are contributing to something we need, right? Because you have the resources. But what we can contribute to what you might need would be the philosophy and the approach, may not be the content. How do we apply that to your context? I think that would be the real game changer. Absolutely.
Thank you. My other audience, please.
There's a question in the chat box. Should I read it?
Please read.
Yeah, just read it out.
Yeah. So this is a question from Gaurav, and I'm just going to paraphrase it a bit. So he's asking, creative pursuits often are less rewarding compared to the inputs they demand, and how could we address that?
Okay. So, see, a couple of things. The question is, what do we mean by creative pursuits? And what do we mean by rewards? Okay, rewards mean different things to different people. Now, when we talk about a creative pursuit, many of us seem to imagine, as I used to imagine, I used to think, well, I'm a banker and I'm going to move up the bank, and there's an artist who has a creative pursuit, or somebody else, a sculptor, and, you know, fine, that's what they want to do. But actually, it's not that. The question really is, how can you bring in creative thinking into what you're doing? So you may be a banker who's not particularly creative or innovative. By the way, we don't want bankers to be too creative or innovative. So probably bad example. But you may be an employee in a company or a businessman. You need to make money, or a businesswoman. You need to ask yourself, am I actually producing something that requires creativity and innovation? If not, how can I bring that into my business, into my own commercial model to create something that's unique and different? And if I did that, then I guarantee you long-term, you will achieve your objectives even better. You will earn more money. You know, if you look at some of the top companies in the world, they're all talking about building intellectual capital because they know that unless they did it, they cannot have long-term success. So a creative pursuit is not something in isolation. It's not a hobby. You can have creative hobbies, not against that. The more important point is in what you do you bring a creative bent of mind? That is important. Then you will achieve whatever rewards you seek. Now, many of us might seek psychic rewards. Some want financial rewards. Some want fame and recognition. Some may not want anything. But the point is, creativity and innovation in and of themselves are a way of life. They will enrich your life. So for instance, if you look at ha, 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 this is not some simple reductionist idea. They are sounds, but they're extremely profound ways of living, not just working, but in how you live.
Hi, Peter. May I make a quick comment?
Yeah, please go ahead.
Mr. Ramji, that was really nice, and it's a really nice thing that you're doing for the country. I just wanted to make a comment that actually what I've noticed, when I was in the US, I noticed that a lot of Indian students are way more outspoken than students from other cultures. And even when you come to ICERs, maybe they are just the top 5% or top 2%. They are a lot more vocal and curious and vocal about their curiosity. If you want to add to that.
Yeah, I mean, that's very encouraging. You see, if you're sitting in America, if you're an American and you tell the American the Indian education system is lousy, he or she is going to say, what the heck do you mean? The brightest kid in my daughter's class is an Indian, or Sundar Pichai is head of Google, or Satya Nadella is head of Microsoft. So that is true. That is true. But it is like this, and we discussed this in the early stages of Agastya. Do you want to fill a glass with water, or do you want to raise the level of the ocean by a millimeter? Now, the Sundar Pichais, the Satya Nadellas, and they're at extreme, but there are a whole bunch of doctors, scientists, all of them are all part of the glass filling phenomenon. Okay? The mass of Indians are the ocean. That needs to move up. If you did that, then what you're going to have is you won't have one Sundar Pichai because it's a probabilistic thing, a hit ratio. You might have 100. Okay? So that is one thing. The second thing is, I'm glad to hear that the Indian kids are, by nature, Indians are talkative. So years ago, somebody told me that the world's best conversationalists are French people and Indians. It is a skill we have. We're very good at that. So I'm not surprised when we are in a positive environment, when you're encouraged to open up and speak, we come to the fore. It's not a surprise.
Sir, I can't resist asking one more question.
Yes.
You said as the children spend more time in structured curriculum institutes, they lose the ability, they lose the craving to ask questions, which I completely agree with. What are the components who are contributing to this dip in curiosity level? How to curb them?
Yeah. So I think, you see, at the very base, psychologically, because all problems at the end of the day, there is a psychological reason. And I'm not a psychologist, but I've read a bit of history. And basically, what's happened in India is the sense of fear. Okay, fear is at the root of this. So the question is, fear manifests itself in different ways. You don't question the order. You don't question your parents. You're afraid of being humiliated. So it could be at multiple levels. The question is, why did this fear happen? Probably during the golden age I was referring to, people were less fearsome. Okay, probably. But there have been historical studies that show that when nations suffer massive defeats, I recently read a study about Iran and the effect of the Mongol invasions on Iran, how it destroyed society, that they're still paying for it today, 8 centuries later, because it creates a sense of fear in society. Don't question, don't ask. The mother will say, there's a dictator, be careful. Your father's head may be chopped off. And gradually it becomes the culture. That fear over time leads to what psychologists call learned helplessness. You begin to believe that you can't change things. It's not karma. Karma is a very positive thing. If you do something great, you'll be rewarded. You can't get a better incentive than that. Learned helplessness is, kuch nahi hone wala hai. I can't change. This is my lot. That thinking has become embedded. When that thinking gets embedded, then what happens is it begins to manifest in various institutions in the form of mediocrity, in the form of lack of questioning, innovation, creativity. So really the central task, even for an organization like Agastya, is not to teach more science. That's a medium. It is, can you bring about a shift from learned helplessness to learned optimism? That is the story of Uma. When she said, I'm not afraid to speak anymore, that's at the heart of it. So we need to ask ourselves, we compartmentalize ourselves. We say, the school is lousy. It doesn't encourage questioning. Well, then you need to ask the parent, when did you last ask your child who came home from school, not what grade did you get in class, but what questions did you ask in class today? Chances are very few of us do that. So as parents, we have to change for the schools to change.
Yeah, thank you. Thank you, sir. So Dr.
I have a
Annapurna,
quick question.
Yeah, please go ahead. Yeah.
I have a quick question. Would there be a trade-off between creativity and gaining credits or grades? We are all after scoring or, picking the top performing students, and this trade-off might, lead to loss of creativity. What do you think about it?
Yes, because the current system, at least till recently, has been so focused on grades, because that's something we're comfortable with. It's our comfort zone, right? If you went out and did something and asked a question or tried to do something different, the chances are you will be shot down, penalized at worst. At best, your grades might suffer because you're spending less time on grades. So that is a problem. So this is where the system, at least at the policy level, is beginning to show more tolerance and kindness to people and saying, let's lower the workload. Let us have a more broad-based way of measuring performance. It's not just grades, it's other things. Now, this transition is not easy, but it's possible. While we are going through the transition, we are going to have to go through such difficult periods and questions. But since, we are doing all this in memory of C.V. Raman, who always topped his, his tests and class, he famously said, throw the textbooks out the window. Okay, so yes, we have to take some radical measures, but there has to be a transition. It's not an easy thing. There will be trade-offs in terms of the old metrics as we move to the new metrics, yes. But institutions like ICER and IITs are beacons. If they took a view that we're not interested in grades, for example, we have a different way of assessing prospective candidates, then that signal will seep down very, very quickly through the system, because everyone dreams of getting into an IIT or an ICER. So if your dream says, I've changed the metrics, then you don't need to convince me. I will blindly go for the new metrics. So that is the responsibility of institutions like IIT and ICER and other similar institutions. They have to signal a change.
Thank you, sir. So with this, I would like to ask Dr. Nirmala to give her vote of thanks.
Good afternoon, everyone. I'm sure you'll all agree with me that it has been a wonderful session and a great start to our own Science Day celebrations here at ICER Tirupati. First and foremost, I would like to thank our speakers for today, Dr. Jameel and Sri Ramji, for being with us today here and sharing their thoughts and experiences. Dr. Jameel, you shared with us an amazing overview of COVID-19, not just its biology, but the number of cases, vaccines. But above all, what struck me was your emphasis on the role of masks and the precautions we all need to take, as we're still not done with this pandemic. So thank you, sir, for a wonderful talk. Sri Ramji, you gave us a wonderful insight into curiosity, creativity, and experiential learning. I'm sure we're all going to think more about aha and aha moments that you described for us. It was also amazing to see the role of the Agastya Foundation to promote innovation at the grassroots level that I'm sure will leave an indelible mark on the nation. So once again, on behalf of ICER Tirupati, we would like to extend our sincere thanks to both of you, and hopefully we'll get to see you actually on campus sometime in the future. I will also take this opportunity to thank our director, Professor Ganesh, for all his support and guidance. And I also want to thank Professor B.J. Rao for conducting this session for us today. I would also like to remind all of you that there is a part 2 for this session today. We are conducting a virtual Science Day presentation for students selected from various schools and colleges through the country. Your presence would be highly appreciated and will be a source of encouragement for these young minds. With that, I would like to close this morning session with a heartfelt thank you to all of you who could join us. And I really hope to see all of you on the other side of the lunch break. Thank you. Thank you.